Dear students, today we shall be talking about osmoregulation in fishes. This is a part in continuity with our earlier lecture, that is structure of kidneys in fish. And that was lecture number 28. So we shall be continuing with now function, that is osmoregulation. First of all, we shall be knowing that what osmoregulation is. When we say osmoregulation, it is a process that regulates the osmotic pressure of fluids and electrolytic balance in organisms. So the process by which the osmotic pressure of the fluids and electrolytic balance is maintained or regulated, that very process is called as the osmoregulation. In animals, this process is brought about by the osmoreceptors. There are various osmoreceptors which detect the osmotic pressure and thus act accordingly. Human and most other warm-blooded organisms, they have osmoreceptors in hypothalamus, and besides the brain, the osmoregulators are also present in kidneys, and that is why they know how to that if there is a salt level is low or salt level is high, and thus they act accordingly to gain the salt or to lose the salt. Now let us talk about the osmoregulation first. That osmoregulation is physiological activity for proper maintenance of water and salt balance of the body tissues in fishes, whether they live in fresh, marine or brackish waters. Or we simply define osmoregulation as a process by which the organism regulates water and electrolytic balance. So when we say water and electrolytic balance to maintain homeostasis, it is active regulation of osmotic pressure of organisms' body fluids which are detected by osmoreceptors. And osmotic pressure is measured. What is then osmotic pressure? When we say osmotic pressure, it is the measure of the tendency of water to move into one solution from another by the process of osmosis. So by the process of osmosis, when the water molecules move into one solution from another, this is called as the osmotic pressure. The higher the osmotic pressure of a solution, the more water tends to move in. So if the solution A has the more osmotic pressure, it means it will gain water. And secondly, when we say osmoregulation, osmoregulation is the active regulation of osmotic pressure of an organism's fluids to maintain the homeostasis of the organism's water content, and that is it keeps the organism's fluid from becoming too diluted or too concentrated. So actually we have to maintain the homeostasis, that is the internal body environment. So how that very internal body environment is maintained? It is maintained by the balance of the salt solution. So that is why we say this very as the homeostasis, wherein organism keeps the fluids from becoming too diluted or too concentrated. So when we say osmotic or osmosis, it is actually a physiological activity very closely related to their survival. Kidneys and gills, they perform the function of osmoregulation, and also in addition they do excrete the nitrogenous waste. So mainly in some cases kidneys, and mainly it is in some cases gills, and mainly it is the kidneys which help in the excretion of the nitrogenous waste and also elimination of the salts, and thus they act as the osmoregulatory organs. So kidneys and gills, they perform the function of osmoregulation, and also in addition many times they are involved in the excretion of the nitrogenous waste. Now let us understand what actually the osmosis is in the case of fishes. So in case of fishes, osmosis tends to promote the loss of water from the body of a marine fish. Now keep in mind that these are the two things you have to keep in mind, that osmosis tends to promote the loss of the water from the body of a marine fish and absorption of the water by that of a freshwater fish. So what happens in this very case? The freshwater fish which is hyperosmotic, the marine fish it is hypoosmotic in relation to its environment. So what will happen? So in case of marine fish, the water will tend to lose from the body. What happens in the case of the freshwater fish? Water will be gained. Now, for example, we do understand this by an example which we have studied in our 11th or 12th class, that for example, if we do have a jar which is separated by a semi-permeable membrane, and this is side A and this is side B, and the equal amount of the water is on both the sides. But when we add a pinch of the salt, in this very side, that is we add salt, the water will tend to move from B to A, and thus the size level of the water will increase in A and it will decrease in B. So what will be the level of the water? The water of the level will be more in A and it will be less in B. So what happened? By the addition of the salt inside A, the water moved from the B side to A side. Now, on the other hand, if we know it in a different way, that the water molecule will move from its higher concentration to its lower concentration through a semi-permeable membrane. Now, you can understand that in the case of the marine fish, the water outside is more salty than that of the inside. So what will happen? The water from the marine fish will tend to lose toward the environment, toward the sea. And what will happen in the case of the freshwater? In the case of freshwater, the body fluids, they are hyperosmotic in comparison to the environment. So what will happen? Water will be absorbed in case of the freshwater fish. So you have to keep in mind, in case of the freshwater fish, there is a water gain. In the case of marine fish, there is a water loss. Now again, what these two fishes have to do? In case of the freshwater fish, it has to conserve the salt now. It will release the dilute urine. On the other hand, this marine fish, as it will take water to compensate the loss of the water, the more salts will get into it, so it will release the concentrated urine so as to maintain the osmotic balance. So this is the important thing. Now, for example, we do have in many cases, mucus in the skin tends to slow the process, but it is not sufficient barrier to prevent the movement of the fluids through the semi-permeable skin. So skin thou acts as a barrier, but it is not a complete barrier. It will also, the water will tend to lose or tend to gain through this very permeable membrane. So when two solutions on two sides of a permeable membrane have different concentrations of dissolved substances, water will pass through the membrane into more concentrated solution. So I told you that this is the more concentrated solution now, where we added the salt and it became concentrated. But this is less concentrated, so water will move from the lower concentration to higher concentration. But talking on another side, the water molecule will move from its higher concentration to lower concentration. But on the other hand, on the other way, we will understand in the way that water molecule will move towards the higher concentration solution, where there are more salts. So again, let us repeat. I will repeat this sentence, that when solutions on two sides of a permeable membrane have different concentrations of dissolved substances, water will move through the membrane into more concentrated solution. So while the dissolved chemicals move into the area of the lower concentration, that is how the diffusion takes place. These organisms in both aquatic and terrestrial environments, they must maintain the right concentration of solutes and amount of water in their body fluids. So this involves excretion. What is excretion? Getting rid of the metabolic waste and other substances such as hormones that would be toxic if allowed to accumulate in the blood via organs such as skin and the kidneys. So keeping the amount of the water and dissolved solutes in a balanced way is referred to as osmoregulation. So the nature of osmoregulatory problem is quite different in different groups of the fishes which live in different environments. There is always a difference between the salinity. There is always a difference in the salinity, what we talked about, the salinity of the fish's environment and that of their inside body. So as the fishes live in different environments, they have to face different environment. Sometimes it is salty, sometimes it is fresh water having very less salt, and sometimes it is brackish water having the moderate amount of the salt. So it depends that how the fish is going to maintain its salt concentration. So it depends whether the fresh water, so they have a different strategy whether the fish is a freshwater fish or a marine fish. So regardless of the salinity of their external environment, fish use osmoregulation to fight the process of diffusion and osmosis, and thus they maintain the internal salt balance of the salt and water, which is very much important and essential to their efficiency and survival. So kidneys do play an important role in osmoregulation, but overall extrarenal mechanisms, they are equally more important sites for maintaining the osmotic homeostasis. When we talk about, there are certain extrarenal sites, they include the gill, tissue, skin, alimentary tract, and the rectal gland and the urinary bladder, which are also involved in the osmoregulation. So actually we talk about the kidneys only, that the kidneys play an important role in the excretion and osmoregulation. But in order to maintain the osmotic homeostasis, there are certain extrarenal sites as well, which I told you that they can be gills, they can be gills, they can be gills, they can be skin, they can be skin, they can be the alimentary tract, they can be rectal glands, they can be urinary bladder. So all these very act in a proper way at the time whenever required for properly maintaining the homeostasis of the fishes. Now let us have a before moving in, let us know the types of the regulation. There are two types of the animals based on how they maintain the internal osmosis or internal homeostasis or the internal environment. So we categorize them as the osmoregulators or we categorize them as the osmoconformers. So osmoregulators are those very organisms that actively regulate, there is a term, actively regulate their osmotic pressure independent of its surrounding environment, that whatever the surrounding environment may be, they will regulate it accordingly, actively regulate it accordingly. And the most freshwater fish are considered to be as the osmoregulatory. But there is second term, we call them as the osmoconformers. So there are organisms that try to match the osmolarity of their body to their surroundings. So what do? They try to have the osmolarity of their body similar to that of their surroundings. So when they conform, that is when they try to simulate the osmolarity of their body with their surrounding, we call them as the osmoconformers. So they conform through active or passive means. Most marine invertebrates, starfish, jellyfish, lobsters, we call them as the osmoconformers. Now let us talk the how freshwater fishes osmoregulate. And keeping in, let us have a little bit that the osmotic pressure of the body fluids of the freshwater fishes, it is always higher than that of the surrounding water. So what does it mean? It means the body fluids of a fish that is hyperosmotic. It means salt concentration inside the fluids of the freshwater fish, it is higher than that of the surrounding. And what happens then? The later diffuses into the body through the oral membrane, gills, and even intestinal surfaces. So that is why the water enters the body through the gills, through drinking, and by the absorption by the skin as well as by the intestinal surfaces. So water tends to get into the body of the fish because it is hyperosmotic. So since the surrounding medium has the low salt concentration, they are faced with the disappearance of their body salts by the continual loss to the environment. So what is again another problem? That as the surrounding medium has the low salt concentration, they are faced with the disappearance of their body salts. So the strategy to safeguard the loss of the salts, what do? They excrete the dilute urine so that the salt is retained inside the body so that their osmotic balance is maintained. Animals such as invertebrates, fishes, amphibians, reptiles, and mammals that live in fresh water, we call them as the osmoregulators. And osmoregulators face two problems. Number one, it is the prevention of the water loss from the body and prevention of the salts from diffusing into the body. So what problems they do have? They have two problems. The one is the prevention of the water loss from the body, and secondly, prevention of the salts diffusing into the body. So two things. And their body fluids, that is 1/3 of the concentration of the seawater, have greater concentration than their surrounding environment. That is why we call them as the hyperosmotic. And as a result, they are constantly taking in water by diffusion through the skin and to much larger extent through the thin membranes of the gills. And the skin has the low permeability to water and the salt ions, hence very little is allowed to enter the body or salt to diffuse out through it. So to counter the continuous inflow of the water through the gills, fresh water produces large amount of the dilute urine. That is what I was telling you. So in case of these very freshwater fishes, the kidney has large number of the well-developed glomeruli. They have the well-developed glomeruli, and more often more than 10,000, which are well vascularized continuously, which you are seeing here. They have the large glomerulus and they are well developed and more in number, more than 10,000, which are well vascularized, and continuously they filter excess of the water which is eliminated as hypotonic, that is the dilute urine, by these very freshwater fishes. So water excretion is the main function of the kidneys in these very fishes. So in the case of freshwater fishes, the water excretion, we say water excretion, it is the main function of the kidneys. But the small quantity of the nitrogenous waste or nitrogenous compounds which contain creatinine, amino acids, ammonia, and urea, they are also present in the urine. So the kidneys of the freshwater fishes is often larger in relation to the body weight. So we have to keep in mind that in relation to the body weight, the kidneys of the freshwater fish, it is larger because they have to function in order to excrete or maintain the water balance by the secretion of the water. And the kidneys of the freshwater fishes, as I was telling you, it is larger in relation to the body weight than that of the marine fishes, and produce copious urine. That is, produce very much amount of urine, which is 5 to 12% of the body weight per day. So almost 5 to 12% of their body weight, they secrete the urine, they excrete the urine. And the freshwater fishes drink very little water and take in little water in their food. So these very freshwater fishes, they take in very less water. They take in the water which is taken along with the food, otherwise they themselves do not take the water. And this very water, as a large amount of water enters into the body of these very fishes by osmosis, and it is more necessary than for the fish. So that is why, as the water is gained by the gills, by the intestine, by the food they eat, the water is gained by the body. So that is why they drink very less water. They prevent water from being drinking. So let us see here that when they take the food and the fresh water, the gills, they actively absorb the sodium chloride, and the water enters osmotically through the gills, and the kidneys excrete dilute urine. It is the intestinal waste and the urine, and then it is the active tubular reabsorption of the sodium chloride so as to prevent the loss of the water. So freshwater fishes lose these salts, especially chlorides, by passive diffusion through the faces. Many times what happens in the case of freshwater fishes, they lose salts, especially chlorides, by the passive diffusion through the membranes. Some salts are lost through the faces, also in urine. Urine also contains some nitrogenous waste, as creatinine. I was telling you about the creatinine, amino acids, urea, and a little bit of ammonia as well. That the freshwater fishes must prevent the net gain of the water. So they must prevent the net gain of the water and the net loss of the salts. So what they have to face, they should restrict the entry of the water, and they should also restrict the loss of the salts. So net intake of water is prevented by kidneys, as it produces more copious or plentiful dilute urine. And it has also been calculated that the freshwater teleosts produce urine which is normally 5 to 12 or 5 to 10% of the body weight per day. The quantity of the salt lost in a day, it varies from different species. It varies from species to species. For example, in the case of salmon, the loss of the water, it is almost in the salmon, the loss of the salts may be up to 17%. That is, they lose up to 17% of the salts of the body chlorides, while in the case of goldfish, that is the cariaceous, it is only 5%. So in the case of salmon, it is about 17%. In the case of cariaceous, it is only 5%. And in the proximal and the distal part of the renal tubule, active reabsorption takes place, and in such the loss of the salt is minimum in freshwater fishes. So how do they prevent the loss of the salts? That is, in the proximal and the distal part of the renal tubule, they actively reabsorb the salt, and thus they maintain the salt balance. So this is accomplished mainly by the chloride cells, many often. In the gills, this is also accomplished by the chloride cells in the gills and oral membranes, that is mouth. The salt ions absorbed by these special cells at the base of the gill lamellae, they are many a times the lithium, calcium, chloride, sulfate, phosphate. So these are also prevented from being lost by the gills as well. So cut. So the loss of the salts is, however, kept at the minimum by active resorption in the proximal and the distal tubules, and the loss of the salts is partly compensated through the food and by the absorption of the salt ions from the surrounding water. And this is accomplished mainly by the chloride cells in the gills and oral membrane, that is mouth. The salt ions absorbed by these special cells at the base of the gill lamellae, they are lithium, calcium, bromide, chloride, sulfate, phosphate, and the entire mechanism of the salt balance is under the hormonal control also. The freshwater fishes have remarkable capacity to extract sodium and chloride through their gills from the surrounding water, having less than 1 millimolar that is sodium chloride. And even through the plasma concentration of the salt, it exceeds 100 millimolar moles per liter of the sodium chloride. And thus the sodium chloride actively transported in gills against the concentration gradient, that is 100 times. And in these very fishes, the salt loss and the water uptake is reduced by the integument, and it is considerable with low permeability or impermeability to both water and salt, and also by not drinking water. So that is how these very fishes tend to osmoregulate, tend to maintain the salt water balance. Now let us talk about the osmoregulation in case of marine fishes. Marine fishes, they have a different problem. The marine fishes experience the problem which is just opposite to that in comparison to the freshwater fishes, because their internal salt content, it is less than that of the surrounding water. So marine teleosts, what happens in the case of the marine teleosts, they are hypoosmotic, that is their body fluids is 1/3 of that of the salt water, and they live in a medium which is having higher concentration of the salts, that is the medium is having higher concentration of the salt than that of their body. So the skin of the marine fishes, as in the case of freshwater fishes, is impermeable, and osmotic exchange occur through the gills and the oral membrane. So the talking about the skin of the marine fishes is impermeable, and osmotic changes occur through the gills and the oral membrane. Or in other words, marine fish have the internal osmotic concentration lower than that of the seawater. So it tends to lose water and gain salt. So here the problem is they tend to lose water and they tend to gain salt. So it actively excretes salt out of the gills. So here the problem is opposite. They tend to excrete the salts out from the gills. So marine fishes are faced with the problem of the continuous loss of the water and the gain of the salt. So the they are faced with the continuous loss of the water, that is their body tends to lose water and their body tends to gain water. Now they have to work opposite to it. So they have to prevent the loss of the water, and they have to prevent the gain of the salts. They have to prevent the loss of the water and they have to prevent the gain of the salts in order to maintain their osmotic balance. So to prevent the osmotic dehydration or to maintain their water balance, marine fishes drink large quantities of the seawater. As in the case we talked about the freshwater fishes, they do not tend to drink water, but the water gets in the form of the food, they eat, or through the gills, through the intestinal membrane, through the skin. But in this very case, what is happening? It is opposite. They prevent osmotic dehydration or maintain their water balance. These marine fishes, they drink large quantities of the seawater, but this further increases the salt content of the body. Now, as they face the problem of losing water, in order to compensate that very loss, they drink water. So by drinking water, what is happening? They are adding salt into the body. So now they have one problem, they have a solution, they have another problem, that is they are facing the problem of losing water by drinking water, but by drinking water they are adding salt to their in their body. So what they have to do, they have to excrete the concentrated urine. That is why they excrete the concentrated urine. So that I shall be talking about, that the urine output is considerably reduced to conserve the water. The glomeruli, they are reduced or become non-functional. The distal segment of the tubule is absent. Thus only a small amount of concentrated urine is produced and most of the water is absorbed in the tubule. So what do? In order to conserve water, they excrete very less urine. Now in order to get rid of the salts, they excrete the concentrated urine. So the urine is less, but it is concentrated. So modern bony fishes, that is the marine turtles, they have the body fluid hypotonic to the seawater, so they have the tendency to lose water to the surrounding, particularly from the gills via epithelium. The lost volume of the water is replaced by drinking salt water. About 70 to 80% seawater containing sodium chloride and potassium chloride, it enters the bloodstream by absorption across the intestinal epithelium. And the excess salt absorbed along with the seawater is ultimately received from the blood with the help of the gills by the active transport of the sodium and chloride and sometimes potassium and eliminated into the seawater. Therefore, however, the divalent ions, they are secreted into the kidney. And in some marine fishes, the concentration of the body fluid and the marine water is almost similar. Therefore, they do not require much energy for the maintenance of the osmolarity of their body fluids. Some they have to work hard, and in some they maintain the osmosis or the internal environment of their body in accordance with that of the environment, and thus they do not require much energy for the maintenance of the osmolarity of their body. So thus, in case of marine fishes, the urine is isoosmotic to the blood, but rich in those of the salts such as magnesium, calcium, sulfate, which are not secreted by the gills. So combined osmotic action of the gills and the kidneys in marine teleost resulted in the net retention of the water, that is hypotonic, both to ingested water and that of the urine. So what happens, for example, here in what you are seeing, that the marine fish, it drinks lots of the sea water. Why? Because there is the loss of the sea water through the body. So another important, the salts are getting in. Salts are also getting in. Now they have to get rid of the salts. What do? They urinate, but urinate very less in order to conserve water, but urinate as the urine is concentrated. Sharks, skates, and rays, they maintain the internal salt concentration, that is 1/3 seawater and the remaining 2/3 is urea. And the trimethyl amino oxide, we call it as a TMAO, trimethyl amino oxide. So the total internal osmotic concentration equals to that of the seawater. So some fishes such as sharks, skates, and rays, they retain the urea and trimethyl amino oxide in their bloodstream, so that their tonicity is increased, and thus they resemble, its osmolarity resembles to that of the external environment, and thus they maintain the osmotic concentration. Now talking about again here, that the gill membrane has low permeability to urea. You have to keep in mind that the gill membrane has low permeability to urea, so it is retained by the fish, and because the internal inorganic and organic salt concentration, they mimic that of their environment, that is the passive environment, influx or efflux is minimized. And retention of the TMAO, these fishes, they retain urea and TMAO in the bloodstream by reabsorption of these solutes from the urine filtrate and by the special segment of the urinary tubule, and thus they maintain their osmoregulation. The secretion of the excess salt by the rectal glands. The gills of these very fishes, which do not have the special salt secreting chloride cells, and although some salts are excreted in urine, but kidneys are not the major source of the excess sodium chloride secretion. So in order to maintain the salt balance, what happens? The rectal gland, they secrete the fluids containing higher concentration of the sodium chloride. So in those very fishes in which the gills, they are not capable, they are not specialized to secrete these very chloride cells, or they do not have the chloride secreting the chloride cells, what do they do? And the kidneys, they are not the major source of the excess sodium chloride secretion. In such fishes, the rectal glands, they secrete the fluids containing higher concentration of the sodium chloride. Then the classic example is hagfish, the myxine. whose plasma is isoosmotic to the environment. So what myxine does, that it makes its internal environment similar to that of the external environment, thus maintaining the isoosmotic balance. The hagfishes, they maintain the concentration of the calcium, magnesium and sulfate significantly lower, and sodium and chloride higher in comparison to the seawater, and thus they maintain their internal osmotic balance. Now let us talk about the control of the osmoregulation. How osmoregulation is controlled. The concentration and dilution of the urine is controlled by hormones, which affect the rate of the renal filtration. So it is the hormones which affect the rate of the renal filtration by changing the blood pressure and thus control the quantity of the urine. So it is the hormones which affects the renal filtration it by changing the pressure of the blood, and thus they control the quantity of the urine to be released. The hormones also influence the rate of the diffusion and absorption across the gill epithelium. So it is the hormones which also control the quantity of the urine, and secondly they influence the rate of the diffusion and absorption across the gill epithelium, and thirdly the thyroid gland and the suprarenal bodies, they secrete adrenocortical hormones. Which control osmoregulation in fishes. In case of anguilla, that is eels, so adrenaline has been shown to have a strong vasodilatory effect on the gill vessels. It reduces or completely stops the chloride secretion. And in the case of salmon, it has been seen that hypothalamus and gonads, they have been implicated in the process of the mineral balance. So in the case of salmon, hypothalamus and the gonads have been implicated in the process of the mineral balance as well. Now talking on lastly, that what is the importance of the osmoregulation. Osmoregulation is a process that requires expenditure of much energy on part of the fish, even when they appear to be inactive. So it seems on the externally, it seems that the fish is inactive, but inside it the active regulation is going on, and they are expending much amount of energy to get rid of the salts or to maintain the osmotic balance of the body. So this constant expenditure of energy to maintain the osmotic balance is a reason why proper nutrition and the low stress levels are important for the healthy fish. So that is why it is stressed upon the aquaculturists that you should feed properly, you should feed the fish properly, and there should be low stress in terms of the environmental factors to the fish, so that you may be having a healthy fish, or in nature as well. So damage to the kidneys through the bacterial infection or other means is often deadly, and these organs extract the large amount of the water which continuously diffuse into the fish body. So damage of the kidneys through the bacterial infection and other means, that is also deadly often, as these organs extract the large amount of the water which continuously diffuse into the fish body, and thus the osmoregulation is disturbed, and that is why it is very much harmful for the fish. So this is all for today's, that is the osmoregulation in case of fishes. And today we talked about the osmoregulators, where in the certain marine and the freshwater fishes, they actively maintain the salt water balance, whereas there are certain which increase the concentration, they increase the concentration of the fluids inside their body, so as to have the similar concentration with that of the environment, and thus they conform to the environment, and they just live in the environment wherein they are required to. So this is all for today's lecture, that is osmoregulation in the case of fishes. Next time we shall be dealing with the nervous system and nervous system structure and function. Thank you very much.